Hello, and welcome to DDW Tech Talks, a series where we examine the latest technologies and services that are helping to drive essential drug discovery research. My name is Rhys Armstrong, I'm the editor for DDW and the host of DDW Tech Talks. Today we're joined by Vikram Devgan, VP of Global Marketing and Product Management at Bruker Cellular Analysis. Bruker has recently launched Beacon Discovery, a platform that enables researchers to analyze live single cells and explore multimodal and temporal cellular responses in real time, linking functional data to sequencing from the same single cell for deeper scientific breakthroughs. We'll now hear from Vikram, who will give a short presentation detailing the applications of Beacon Discovery before we move on to a Q&A. Over to you, Vikram.
Hello, everyone. I'm Vikram Devgan, VP of Global Marketing and Product Management at Bruker Cellular Analysis. I'm super excited to introduce you to our newly launched platform, Beacon Discovery. It's a major leap forward in live single-cell function analysis, delivering the deepest insights into a single-cell behavior. Over the last 2 decades, we have seen remarkable progress in single-cell research. We began by identifying what cell type exists using flow cytometry. Then we moved into molecular state of those cells via single-cell multiomics. Spatial biology helped us to understand where these cells reside and how they are organized in a tissue. But even with all this, one crucial question remained. What are these cells actually doing in real time? That's where Beacon Discovery comes in. It empowers you to capture multiple functional parameters over time on the same single cells. This functional layer is especially critical in fields like cell therapy development and antibody discovery. Before going any further, I would like to share a short video with you. It gives you a feel for what Beacon Discovery looks like in action, its capabilities and potential impact. So let's take a look. Okay, so Beacon Discovery brings together live single-cell culture, deep functional profiling, and real-time data visualization. And above all, we have made it accessible. The cost is significantly lower than our premium Beacon system. It's compact, small enough to fit on a benchtop. It comes with a brand new intuitive user interface to simplify even a complex workflows. And it's built on our proven platform, already trusted by over 160 customers worldwide. Each Beacon Discovery experiment begins with precise automated setup. Using our optoelectric positioning technology, we gently move reagent beads, single-cell under evaluation, and additional effector or target cells into a nano pen chambers on the chip. Then, this will allow you to set up thousands of parallel single-cell experiments with exquisite control. Then you flow in assay reagents like antibodies or fluorescent strain through the channels of the chip and get fluorescent readout over multiple time points using built-in microscope. Now the question is, what assays can you run? For T cells, you can do dynamic cytotoxicity, multiple cytokine secretion, cell-cell interaction, and motility assays. For antibody-producing B cells, you can do antigen specificity, affinity, cross-reactivity, internalization assays. And all of these is measured on the same living cells over time, giving you true multimodal and temporal functional analysis. In fact, most of our users run 4 to 5 assays on the same single cells over 24 hours, capturing readout every 30 minutes. Once you have identified most interesting single cells, you can export them live for downstream sequencing. What you can do for then is you can perform TCR sequencing, BCR sequencing, or mRNA sequencing. This gives you the ability to link function to a transcriptomic data and reveal insights like differential gene expression in highly functional or polyfunctional immune cells, as shown here. Beacon Discovery is already enabling critical workflows in multiple applications, including T-cell profiling and cell therapy development, all the way from TCR and CAR discovery, validation, and biomarker identification. Another major application is antibody discovery, including therapeutic, autoantibodies, and even vaccine development. In summary, Beacon Discovery is truly making functional live single-cell analysis more accessible, and we believe that this will be a big catalyst for breakthroughs in immunology, oncology, and beyond. Thank you.
Well, Vikram, thank you so much for that presentation. I think it was a great overview and very insightful look at the Beacon Discovery platform, and it should be very useful for our audience as well. I'd just like to dive into a few more aspects of the platform, if you don't mind. So we can start with this question. What unmet needs in single-cell research does Beacon Discovery address?
Yeah, thanks for the question. Single-cell research has came a long way, but there are still major gaps, especially when it comes to comprehensive understanding of cellular function in a more dynamic way. So most current tools give you only a snapshot or force you to destroy the cell to get the data. With Beacon Discovery, we are filling that gap. So it actually lets researchers track the same live cell across multiple assays and multiple time points. So what does that mean? That means that you are not just getting a static profile, you are watching more like a video, more like watching a biology unfold in a real time at a single cell level. And this opens the door to understand cellular behavior, mechanism, and therapeutic potential in a much more detail, in a much more depth and breadth.
Amazing, thank you. It sounds so useful. And just moving on to a second question, how does Beacon Discovery enable sequential multi-assay analysis on the same live cell? And what biological insights does that unlock?
Yeah, I think this is really where Beacon Discovery shines. The platform uses optofluidic technology. That's our proprietary technology from Bruker Cellular Analysis. Mm-hmm. And what it does is this technology allows to gently isolate and retain individual live cell in a tiny reaction chambers. And we call these tiny reaction chambers nanopens. And this all is happening while this technology is keeping these single cells healthy and happy. And what it does is that will allow you to run multiple sequential assays on the same cell over time. You can start by measuring like cell surface marker expression, then follow up with multiplex cytokine secretion, dynamic cytotoxicity, and even you can do real-time imaging of cell-cell interaction. And all that is on the same single cell. And this capabilities let you build more like a multimodal and temporal profile of cellular function. And throughout this process, the cells are perfused with culture media, and that will keep them healthy and viable for the long time. And the beauty, I just want to add one more thing on here. The beauty is once you have identified functionally interesting cells, you can retrieve them for downstream sequencing. And that allows you to identify really a rare, highly potent therapeutic candidates.
Great, thank you, Vikram. And just for the next question, in what ways is Beacon Discovery optimized for accessibility for academic and biotech researchers?
Yeah, great question. We are actually listening very closely to what scientists were asking for. They are looking for an accessible and flexible system for live single cell function analysis. And Beacon Discovery does exactly that. Beacon Discovery was designed specifically to meet these needs. First of all, it's a small benchtop footprint. It has a very intuitive, easy to use user interface. And we have streamlined all the workflows on Beacon Discovery. But most importantly, it is priced to be within the reach of academic labs, small biotechs, and translational research groups in pharma. So whether you are a PI in an academic setting or working in a lean biopharma team or have a core lab, now you have access to live functional single cell analysis in a very cost-effective manner.
Yeah, so important for that transition from academia in pharma. And just for another question, what types of discoveries or experimental workflows will be possible with Beacon Discovery?
Yeah, so Beacon Discovery opens the door to a wide range of high-impact research. That's including immuno-oncology, cell therapy development, antibody discovery, or even vaccine response profiling. So let's take an example here. Let's take an example of therapeutic antibody discovery. For this, you can screen thousands of individual B cells to identify rare, high-value antibody-secreting B cells using Beacon Discovery. And because you're not limited to one assay, you can run multiple functional tests or multiple screening tests in sequence, like IgG secretion, antibody binding specificity assays, affinity assays, cross-reactivities assays, or actually you can even dig into you go beyond the binding and look into the real functions like neutralization part of it. And all you are doing on the same B cells across thousands of B cells in a one single day. That's the power of Beacon Discovery. And once you have identified the hits, you can recover those cells, seamlessly obtain BCR sequence, and all using our integrated end-to-end workflow. So at the end, you get the seamless way to get the entire hit to lead process in one place with a one single platform. And overall, Beacon Discovery is designed to be incredibly flexible, and it designed to give researchers breadth and depth in their experimental design.
Great, and that's so important within the lab. Just for a final question then, what impact do you hope Beacon Discovery will have within the next few years?
Yeah, see, our goal is to really democratize this live single cell function analysis. In the next few years, I would like to see Beacon Discovery sitting in most of the labs and becomes a fundamental tool in the cell therapy development and antibody screening part or for therapeutic antibody discovery or vaccine development part of the field. And what we want, we basically want to help really accelerate this therapeutic candidate selection, reduce failure rate, and uncover mechanism that cannot be seen before. Ultimately, it's all about enabling smarter science, right? And making faster progress toward treatments and improve lives. That's what we want to do at the end, right?
Yeah, so important. Well, thank you, Vikram. That brings us to the end of the DDW Tech Talk. So I'd just like to thank you for your time. Thank you for sharing all your insights into Beacon Discovery. And thank you to the audience for watching.
Thanks a lot. Thanks for your time.
Thanks, Vikram. Thank you to Bruker Cellular Analysis for sponsoring this DDW Tech Talks. You can now view Drug Discovery World content online on your mobile or tablet. Thank you for listening.